Thank you.
Okay. So a lot like with T cells, the main goal here is to create kind of a clone of cells that all have a really good specificity for a particular antigen. What's really different about B cells is that their job is to give rise to plasma cells that secrete antibodies, which can bind to particular pathogens or pieces of pollen or whatever is triggering your immune system. Anything that poses a threat to the host. Your antigen, basically. So the main points of this, and some of this goes back to stuff we learned really, really early in this semester. Your immature B cells all have immunoglobulin receptors on their cell surface. We call that the B cell receptor. As they become mature, they'll actually lose expression of those cell surface ones and start making secreted versions. Does anybody remember what causes that change in that process? Splicing. Yes, it is alternative splicing that causes the difference between membrane-bound or secreted versions of your immunoglobulins. Excellent. All the receptors on a single B cell should have identical specificity for antigen. When you stimulate those B cells, when they encounter their specific antigen, they mature and they migrate to the lymphoid organs where they replicate. They're already in the lymphoid organs, but even within those lymphoid organs, they will do a little bit of migration. We're going to look at the structure of lymph nodes, and you'll get to see kind of the pathway that they take and a little bit about why that actually occurs, which I did think was kind of interesting. And then once that cell replicates, that's where that little clone army comes from. And then at the end of the immune response, most of those B cells are going to die off. Some of them will remain as memory cells. And again, a lot like with the T cells, these seem to be generated early during the immune response, like during that proliferative burst phase, and not necessarily the ones that are leftover survivors at the end of the response. So the process here isn't that different than what we saw for T cells. It's just that there's a bunch of extra steps and signals and maturation that really happens that are very unique to B cells that are really, really wild in biology. I mean, this is some crazy stuff that we're not classically taught happens with cells. You know, y'all are taught from the beginnings of biology that all your cells contain the same DNA. And we've already learned this semester that that's not totally true because your B cells edit out chunks of their own DNA to make different recombinatorial B cell receptors. So they're clearly different. And then in this chapter, once they get triggered by antigen, they're going to go through another round of maturing and another round of editing in their genetics. We're not going to go as deep into the details on this level of editing. Yay. This chapter in your textbook was kind of dense, so I'm going to try to keep it a little bit simpler. This is just kind of the starting point. And so that's kind of the graphical overview of what sort of happens is you start off with the hematopoietic stem cells. This part was covered on the previous exam. These give rise through gene rearrangements to small clones of cells that go through a few maturational processes, first in the bone marrow, then in the spleen. And then once they get to the spleen, they sit around and they look for their antigens. And then this is kind of where we pick up their story and how we get to antibody secreting cells, also known as plasma cells. Okay, there's two types of B cell responses. And these are elicited by distinct antigen types. If you're wondering, there are two important abbreviations that you'll see in here because it saved space. AG is the most common abbreviation for antigen. AB, antibody. That will also save you time writing it in your notes. So you have B cell responses that rely on T cells. These are kind of your classic responses, the ones that we learned the most about so far, and the ones that we're going to continue to talk the most about that involve your B2 B cells. And then you have your T-independent responses that do not require help from helper T cells. So one group of your responses is going to require CD4 positive T cells in the lymph nodes or spleen, and the other group of B cells does not, and those are primarily your B1 and marginal zone B cells involved in the T-independent responses. That's TI for T-independent. We'll come back to those kind of at the end of this chapter. So first we get to talk about what actually happens during the response for the T-dependent types, and that's where the bulk of this chapter really focused. All right. So the first thing that has to happen is your B cell has to encounter antigen. This is going to trigger an initial activation and burst of proliferation. Then your B cells are going to kind of split in one of two, or well, basically kind of two directions. Either becoming plasma cells at that stage, or going to the germinal center in the lymph node and becoming what are known as germinal center cells for a little while, before they become really weirdly specific plasma cells. Eventually, they should all become plasma cells or memory cells. So it's this proliferation that actually causes the formation of what are called the germinal centers and the primary foci in the lymph nodes or spleen. We'll come back to those concepts, so don't freak out yet. But this is basically the triggering of this is when these B cells encounter antigen and they process it and they express it on their surface with class 2 MHC. Remember, B cells can also serve as antigen presenting cells. So if your antigen comes in coupled with complement, there's a particular receptor on B cells that's CD21, and it is important for detecting complement-coded antigens. Those are usually going to be bloodborne, so mostly they'll be filtered by the spleen, and this is, again, a concept that'll come up in a minute. Most of what we're going to talk about are the T-dependent responses, so the ones that require interaction with helper T cells. And that's actually where we'll start, with the process of activation. So in the early immunology days, they did some interesting experiments to try to figure out, like, how did T cells and B cells interact, or which ones were responsible for the immune response, and how did this all work? So basically, they irradiated some mice, and then they tried to reconstitute those mice with various different components of the immune system. So nothing, those were negative control mice. B cells only from the bone marrow. Thymus cells only from the thymus of a non-irradiated mouse. So just T cells, or a combination of B cells and T cells. And then they looked for which mice could actually make antibodies. So which mice had a functional B cell response. And the only group were the ones that also had the thymus cells, the T cells. So this told researchers that T cells are actually necessary to get good production of antibodies. You can get some production of antibodies without that, but it's a weaker response, and we'll talk a little bit about what that response looks like too. So things you should remember from previous stuff. B cells come from the bone marrow, T cell, so they are called B cells. T cells come from the thymus. That's pretty normal. Recirculating B cells, interestingly enough, have a half-life of about 4.5 months. So from the time they actually reach the spleen, they should stick around in your body for around 4.5 months looking for antigen. That's their half-life. Some will live longer than that, others will live shorter than that, but that's about where half of them will have disappeared. They'll have been replaced with other things. So, fairly large figure here, but there are three main fates of B cells following antigen stimulation. This is for the T-dependent pathway. They can proliferate and form a primary focus. These cells are going to start secreting IgM. So they will switch, because of alternative splicing, from making membrane-bound IgM to making soluble IgM. They will begin secreting the IgM antibody. Or they can become memory IgM cells. Or they can enter the germinal center, where they undergo two processes. We'll learn about these in more detail. Somatic hypermutation and affinity selection. Big words. Magic happens. Germinal center. Good. We'll figure out what that magic is shortly. Probably not all of it today. So what's exactly going on in this particular figure? This is mostly kind of the visual summary of some of that stuff. So this is when you get interaction between the B cell, and you've got B cell receptor signaling, you've got some cytokines that are coming in, and you've got interactions with a helper T cell. This triggers one of these three main pathways: entering into the germinal centers, becoming memory cells, or forming a primary focus. This bottom part of the figure down here, this organ is supposed to represent one of your secondary lymphoid organs. This is, let's call it the spleen in this particular case, since that's where we deal with a lot of the B cell development and stuff. You've got your thymus up here and your bone marrow up here. These contribute the cells that populate this. And then you get antigens that come in from one of two sources: the lymphatic fluid or the blood. So, blood-borne antigens are filtered by the spleen. That part is important. Tissue antigens that are like in your skin tend to get drained by the lymphatic fluid in your body, like the interstitial fluid between your cells and your tissues. That gets drained back into your lymph nodes. So, blood-borne bacteria, viruses, etc., those get filtered by the spleen. The ones that are just in your tissues, like normal mucosal tissue, etc., those get filtered by your nearest lymph nodes. The B cells first have to acquire their antigen. So there's a few different ways that they can get these antigens. They can drink it directly from the surrounding fluids. This is through penocytosis, drinking small amounts of extracellular fluid. Penocytosis is a pretty crummy way to acquire antigen, because they'll get a lot of different types of antigens, and most of them won't be specific for that particular B cell's receptor. They can steal antigen from macrophages that are present in the lymph nodes or spleen, or follicular dendritic cells. These follicular dendritic cells, one of the things that we need to realize about them or know about them is that the surfaces of these cells are studded with antigen-antibody complexes that these dendritic cells have picked up, usually in peripheral tissues. So the dendritic cells found these pathogens in the body, and then they migrated to the lymph nodes or spleen, where they're going to show these off and have these displayed as antigen-antibody complexes on the surface of these cells. And the B cells can actually literally steal that antigen right off the surface of the dendritic cell. The mechanisms for that are not as complicated as I thought they would be. So dendritic cells are actually your best reservoir of those antigens, because they can actually hold on to those antigens for weeks, whereas macrophages can only hold on to antigen for a few hours. Again, dendritic cells really are the best antigen presenting cells. They are our key APCs. Okay, so once the B cell recognizes cell-bound antigen by contacting it usually on like a dendritic cell, it'll recognize it through its B cell receptor. It'll then trigger. The B cell is actually going to melt over the surface of that antigen presenting cell. It's literally going to spread itself and just give a giant hug and try to engulf a big section of that antigen presenting cell. It's trying to maximize contact between the B cell membrane and the dendritic cell membrane. The more overlap of those two membranes that you can get, the more antigen and B cell receptors and MHC stuff you can cluster into one area, which is what's going to happen after that. So in this image, what you're actually looking at is that B cell spreading. This is in the beginning, this is the antigen presenting cell. After 2 minutes, this is the ring that you kind of see, or this is the B cell membrane that you see that has contacted that antigen presenting cell. By 5 minutes, it has contracted a fair amount, so it's like shrunk in on itself. By 10 minutes, it's almost completely gone. So this is a really quick process. It's a quick, basically the B cell kind of goes and sucks in as much as everything as it can into these small little areas, and it creates the immunological synapse that way, a lot like the one that we saw with T cells. And that's exactly what we're looking at here. This immunological synapse is a little more complicated than the one that we looked at for T cells, but it's not that bad. So in the center of your immunological synapse, once again, you have, instead of the T cell receptors, these are B cells. So guess what kind of receptors you have? B cell receptors. Yeah. In the center of this, the heart of this, is the interaction between the B cell receptor and the antigen on your antigen presenting cell. You've also got the necessary co-stimulatory molecules, Ig alpha and Ig beta, that are necessary for signaling through that B cell receptor. And you've got some intracellular signal transduction stuff that we'll go over in a couple minutes if we get to that today too. So tyrosine kinases and ITAM regions within the tails of these signaling molecules, which go back to things that we also learned about at the beginning of the semester when we talked about the structure of this thing. So this is the center ring of your immunological synapse. The next layer out, this blue layer, these are integrins, which are cell adhesion molecules. Their job is to act like cellular Velcro and hold cells together. So this ring in particular helps to anchor these two cells together and keep these B cell receptors and antigen portions kind of clustered in one particular area. Usually they'll cluster together into lipid rafts. And then the third layer out, this last ring, is composed of actin. So the cells literally remodel their cytoskeleton a little bit so that you get these kind of processes that extend up from the B cell to physically touch the membrane of the antigen presenting cell. So in T cells, there was actually one less layer. We didn't see that actin layer in particular, but we still saw the T cell receptor and MHC in the center of that, followed by co-stimulatory molecules in the margins, or in the what was called the PSMAC region. It's the same idea here. The central region is called the CSMAC, Central Supramolecular Activation Complex. The outer ring, or the integrin ring, is the PSMAC peripheral. And then the actin ring in this case is known as the DSMAC for distal. The longer these molecules and these cells stay in this configuration, the longer they stay attached like this, the stronger the signaling is going to be. And usually this is driven by how well that B cell receptor can recognize antigen. How good is that match between the membrane-bound antibody and the antigen-presenting cell's antigen? If it's a really strong match, you'll have longer interactions. If it's a fairly weak match, the interactions will be a little bit shorter. And this leads to signal transduction within the cell. It is complex. There are at least 3 families of tyrosine kinases that are involved in these processes. The SRC family kinases, the SYK family kinases, and the TEC kinases. I am not going to worry about you memorizing any of those in particular. I want you to basically have a basic understanding that we start here. There's our membrane-bound IgM. It has recognized antigen. You've got supplemental signaling coming through CD21. You've got activation of these ITAM regions. The tyrosine kinases, Lin and Syk, start off these different signaling pathways and signaling cascades. So you can see that there's at least like 1, 2, 3, 4, 5 6 different pathways that are triggered by this. Cell signaling occurs. Everybody happy with that? Lots of different ways.
Is the only antibody you can, or not antibody, the only BCR that you can have IgM, or could it also be IgG and so on?
IgG is not yet expressed.
Okay.
So right now it's IgM.
You can have IgD?
You may have some IgD as well, yes.
Okay, that makes sense. Just like the naive IGs.
Yep, these are like right at the bridge between becoming a naive B cell and becoming a mature B cell. This is that initial step in activation. So the reason this is so bleeping complex is because the changes that this B cell is about to go through are going to require hundreds of genes to all be regulated at the same time. Some genes are going to be upregulated and expressed, other genes are going to be squashed and silenced. So that's why I am not digging any farther down in these pathways than just, oh look, cell signaling happens. Good, okay, because I don't know what hundreds of genes are being regulated by these processes. Y'all don't need to know that at this stage either. Just that there's a lot of changes in gene expression that's going to happen. Depending on the strength and duration of the signal and the cytokines present in the environment that provide some of those co-stimulatory signals, you're going to get different outcomes of this signaling. So remember, I said that these cells could either become like primary focus cells or they could become germinal center cells. So this is one of those two things, like how long these things interact with antigen and how long you get cell signaling going on and what types of cytokines and co-stimulatory signals you receive direct the cells to one of those two fates, primary focus or germinal center. So how does the B cell acquire its antigen? I always wondered this. This was so cool. I liked reading this part. There's essentially 3 methods for B cells to get antigens from their environment. So remember, they're one of the 3 main types of antigen-presenting cells. The other 2 are dendritic cells and macrophages. Good, you guys have learned some stuff. The first method is just by sampling and penocytosis of that lymph fluid that's around them. Just the extracellular fluid. Most cells do this. They constantly drink a little bit of the extracellular environment. The cells that are the best at this are the dendritic cells. B cells do it too, and they will acquire some of their antigen that way. But penocytosis basically sucks because they're going to get a mix of antigens and limited quantities of them. Like it's going to be really dilute. what are the chances that that particular B cell is going to pick up on that particular virus, as opposed to all of the other little proteins and peptides floating around in that fluid? Low probability. So the other two mechanisms are going to have much better results. And one of those, both of them involve basically stealing the antigen from an antigen-presenting cell. So B cell is going to get right up next to that antigen-presenting cell, and that's going to recognize, you've got your membrane-bound receptor here. It's going to actually recognize the antigen on the antigen-presenting cell, and there's one of two options. The B cell can, one, secrete lysosomes into this space between the cells. The lysosomes dump their contents and literally digest the antigen right out of the MHC. That's cool. It's like a Venus flytrap. It secretes the enzymes that digest its food outside of its own little planty body. This is the same kind of idea. The digestive enzymes get dumped between the cells and literally strip the antigens off the surface of the antigen-presenting cell. The APCs have plenty of that anyway. Option two, if the interaction is strong enough between the B cell receptor and the antigen, it can literally rip it out of the membrane of the antigen-presenting cell and steal it that way. Just physical force, just yank it right out of the membrane.
Does it rupture its membrane?
No.
No? It
No.
just like...
Yep.
Does it take the MHC too?
Yes, it takes the MHC also. And then that whole thing gets endocytosed and processed through the normal antigen-presenting pathways. So this would be the endosomal pathway for MHC2 presentation. And then those B cells will re-express that same antigen on their surface with the MHC2. So that's kind of what's happening over in this side of things. You've got antigen recognition. You've got vesicle that's got some endocytosed antigen and membrane from this APC cell membrane. They don't really show the secretion option, though, in this particular figure. Kind of wish they had. But you can see some of the cytoskeleton molecules, actin and myosin, that actually attach to the B cell receptors and help to pull those antigen-presenting cell MHC and antigen complexes. Remember, if we're talking about the follicular dendritic cells, their surfaces are studded with those antigen-antibody complexes anyway. So they've got plenty to spare.
So does this happen, is this the same kind of mechanism as the synapse, or is that like two different things?
This is part of the synapse, yes. This is one of the events that should be going on, and the longer these two things stay in contact, the better and the more antigen that B cell can acquire. So that B cell needs to acquire a bunch of antigen. It's going to need it. And the reason it needs that antigen is because it has to present antigen itself. It has to do its role as an antigen presenting cell and show that antigen to the T helper cells. So the B cell really needs to maximize its intake of antigen, and in particular of the very specific antigen that it can recognize. So it can already recognize and detect this particular antigen. All the other ones it sampled through pinocytosis, maybe not. Like, it doesn't guarantee a good fit. But the thieving process, the stealing it from the antigen presenting cell, this guarantees an excellent fit between the antigen and the B cell receptor. So we know now that the B cell is literally just stealing its own specific antigen, which then it's going to show off to the T cell, which is going to use the T cell receptor, which is also antigen specific. Think about this for a second. You're going to end up with two lymphocytes, a B cell and a T cell on either side of this antigen. Both of them are going to have the ability to recognize that exact same antigen. They pair up. Somehow my mind starts making weird, funny noises at this stage and going, but the probability of that mathematically has to be like so low. Oh my God, how does this even work? You've got millions and millions of T cells and millions of B cells, and they have to find the right antigens and each other in this process? How do we manage to mount immune responses? I don't get it. And you slaughtered a ton of T cells. Okay.
Do we know how it happens? Like how they happen to meet each other in this like very specific manner?
Yeah, I mean, the cells are constantly migrating between lymph nodes to try to look for those antigens. If they manage to find their cognate antigen within that half-life of around 4.5 months, then they can mount an efficient response. If they don't, they'll eventually die of apoptosis.
Wow. So it's literally just like.
It's a game of really random odds.
Do you know what the probability is? Has anyone calculated that?
I'm sure some math geek has calculated the probability of that. I don't know what it is offhand, but I'm assuming it's a number that's so small that it's going to make my head hurt to think about it.
So, like Dr. Gaff could help with that if we wanted to figure it out.
Dr. Gaff could help with that. I mean, it's not terribly difficult math. It's just the probability of 1 in a tiny, tiny number, and the probability of 2 in a tiny, tiny number.
Yeah. Ooh.
And the product of those two probabilities. Yeah. We got to be talking like, I don't know, 10 to the minus 12, 10 to the minus 14, something like that. So once the signaling starts at that B cell receptor, those B cell receptor and antigen complexes get engulfed into the B cell. The B cell eats them, swallows them, and uses that antigen to present back on its surface with MHC2. That's, again, the exogenous pathway, or the endocytic pathway, whichever one you want to call it. I don't care which, as long as you know what it means. And those antigen peptide fragments are going to be presented back on the surface with MHC class 2 to try to get T cells that fit. B cells that have actually taken up their specific antigen from those follicular dendritic cells that have managed to steal antigen are 10,000-fold, not just times, 10,000-fold more effective at presenting antigen to T cells than the ones who get it by penocytosis. So this leads to a really key point. The only B cells that are really capable of presenting high levels of antigen to T cells are the ones that can make antibody to that antigen. They had to have been able to make antibody to that antigen and then have high enough affinity to steal it away from the dendritic cells so that they could present it to the T cells. And the only way that's going to happen is with really high affinity. Other things that are going to happen when this process begins, the B cell is also going to upregulate co-stimulatory molecules CD40, CD80, and CD86. Those are co-stimulatory molecules necessary for activating a T cell. We learned in the last chapter, and it was on the last test, CD28, CD80, 86, that was signal 2. I see nods, meaning y'all remember that. Of course, we did just have that answer on the test like earlier in this class, but still, it makes me happy. All of this stuff that we've been learning is kind of building on itself at this point, so you've got a really good understanding of what's actually happening. Okay, once the B cells process antigen and start sticking it on their surfaces in MHC and doing their antigen presenting role, they also start to express a particular chemokine receptor, that is CCR7. And what that does is it acts as the receptor for two specific chemokines, CCL19, CC ligand 19, and CC ligand CCL21. These are made by T cells. So the fact that these B cells make this receptor means now they can respond to these T cell signals. And these are chemokines. So they're ones that actually cause migration. When the B cell starts expressing CCR7, it can start migrating toward the sources of these two chemokines, or the T cells. So the B cell starts to migrate in the lymph node or spleen toward the T cell regions. Additionally, there's 3 others, CXCR5, EB12, and CCR7, all of the, or 2 others. They also help direct B cell migration to that boundary between the B cell regions and the T cell regions. We're going to look at the structure of a lymph node on this next slide. Like, it's all figures, so that's why there's so much text on this one. So B cells are going to start to move toward those T cells that they actually needed to get full activation. Once they encounter their counterpart that is also an antigen-specific T cell for that same antigen, despite the odds, they get pro-survival and maturation signals, including things like IL-4 and IL-21. And these were 2 cytokines that were characteristic of Th2 or type 2 responses. Oh God, it all fits. Help, my head. Yeah, but I like it when things fit. When the puzzle pieces click, it's very gratifying. It's kind of nice. All right. Once they start getting these 2 cytokines, they downregulate CCR7. This is only 2 to 3 days after antigen exposure. And then they leave the T cell areas. So the B cells at this point, they encounter antigen, steal it away from those follicular dendritic cells, present it on MHC2 along with expressing CCR7. They migrate over, try to find a T cell, look for it for a couple of days, 2 or 3 days. Hopefully they find their counterpart T cell. Then they downregulate that and leave the T cell zone if they find their match, and hopefully enter the follicles. By about 8 days later, no, not even 8 days later, just 2 to 3 days later, the B cells have pretty much migrated back to the middle of the follicle. And there they do one of 2 things. They establish a primary focus and start making IgM, or they start forming a germinal center. And I'm going to talk about all of that on this next slide. I hope I have enough time for this next slide. I know I'm looking at the clock and going, I have like 8 minutes, 7 minutes. Okay, 7 minutes. That's not much time on this. All right, so this is a lymph node or spleen. You've got this kind of lobed structure. You've got basically these nice round follicles that you see around the edges. Your T cell zone is this zone here. So we're talking about like an incoming blood vessel here. You've got your T cells sort of packed in around that. And then your B cells take up residence in the follicles where they wait. So once your B cells get their antigen from the follicular dendritic cells or from other dendritic cells that come into these basically interfollicular zones, like in between the follicles, they get their antigen, and then they're going to migrate toward these yellow regions, in more toward the middle, toward this edge of the follicle. If they find their T cell, they'll then migrate back more toward the middle of the follicle and establish one of 2 places, a germinal center or a primary focus. So that's what we're kind of looking at in this particular set of panels up here. All right, so we've got this yellow area, that's still the T cell zone. This is still basically kind of color corresponding to the figure below it. You've got these light green cells, and this is basically the T and B cell boundary shown in the dotted blue line. These are B cells, and hopefully you get an antigen-specific B cell. In this case, they color-coded that as a darker green. It may be a little hard to visually distinguish, so I'm just going to point at it. This one found its antigen. Oh my God. So it steals its antigen away from the follicular dendritic cell, and it migrates down toward that yellow region, toward that T cell specific zone where it starts looking for its cognate T cell that can also recognize that exact same antigen. If it finds its cognate antigen, ooh, check it out. It multiplies. It goes through several rounds of mitosis. A cluster of those cells basically stays here and generates what are called plasma cells, and this particular point is known as the primary focus. Another group of cells kind of actually migrates further away, back toward where the dendritic cells and the macrophages are going to be, back up toward the purple region. The subcapsular zone is what that's called. Basically, it's just these regions that are like along the edges of the lymph nodes and farthest away from the center. So they're going to migrate back up here, go back and try to get some more antigen, is more or less what they're doing. Assuming they find some, they're actually going to migrate then toward the center of the follicle, and they're going to make their own separate cluster that are the germinal center B cells. And you can see all of this play out in fluorescence microscopy. So, this blue staining, that's B220. That is a B cell specific marker, so we know what we're looking at are basically B cells. The RFP, red fluorescent protein, the red cells in this region, and kind of like you can see this brighter blue band and then these sort of red areas. Those are the T cell zones in red, and then the B cell zones in blue. The antigen-specific B cells show up in green, and so in the naive cell, they're kind of scattered all throughout up there at the top, and there aren't that many of them. Day 1 of antigen encounter, you can see that they actually start to sort of cluster and migrate toward the T cells. By day 2, they're closer to those T cell areas, and you've got better interaction. And by day 3, it's all reversed and gone back the other direction, and there's a whole bunch of them that are now back along the edges of that, but there's a ton more of them. They've gone through a bunch of rounds of proliferation. A B cell can actually replicate about every 6 hours. So that means you can get through 4 rounds of replication in a day. 6 times 4 is 24. First replication, you go from 1 cell to 2 cells, 2 cells to 4 cells, 4 cells to 8 cells, 8 cells to 16 cells. So you should have 16 times more cells by day 3. That's a good start. I want millions because I'm assuming I got swamped by virus and this is why these things are doing this. By day 4, check it out. Another few rounds of proliferation, your numbers are looking much better, and you've now got these bright green areas that are sort of in the middle of these B cell regions and forming what are called germinal centers. Yes?
So when the B cells move to the edge, the B T cell border, they aren't leaving the follicle, right? So are the T cells kind of flowing around so they
the T cells circulate within these T cell zones, and the B cells kind of come up right to the border.
So then as that flow of T cell comes around, that's where they get the chance to actually
So as those T cells circulate through and the B cells circulate through, this is where they kind of get the chance to see if they match with each other, yes.
Okay.
And that turnover and that movement helps to improve the odds that the right T cell is going to find the right B cell. It's still a long shot, but it helps.
If they weren't.
It's better than if they were
if
they were just stationary, then that would never work. Yeah.
Okay, thank you.
All right. I will see you all on Thursday. We will pick up here.